We'll give it one more minute and then we'll go ahead and get started. We've got a good project today.
Mm-hmm.
All right, let me go ahead and start the introduction. So everyone who's on the call today, again, thank you for joining. We have this meeting every 4 months, and it's part of both the Firefighter Cancer Cohort Study and the Firefighter Health Collaborative Research Program that we have here at the University of Arizona, but we invite our really our wonderful colleagues to be able to present at this session. So we're very fortunate today to have Dr. Jackie Goodrich, who's an environmental epidemiologist at the University of Michigan. She's also a co-lead of the Firefighter Cancer Cohort Study and the principal investigator. That means kind of the one who's leading all the efforts for the FEMA-funded high-risk study that's been evaluating exposures in East Palestine, the wildfires in Maui, and also the LA urban conflagrations, and also some additional events, such as one recently with Fairfax County. She's got her PhD in toxicology at the University of Michigan in 2011, and she's had multiple publications over the past, I think, 5+ years on epigenetics and firefighters. So it's really helped push us forward to understand the effects of firefighting exposures on cancer and other disease risks. So, but in addition to that, although we don't hear as much from her about that because she's been really presenting to us on her firefighter work, she's also has expertise in evaluation of impacts of prenatal and subsequent early life exposures on child health and other groups as well. So I'll stop there and I'll hand it over to Jackie.
Well, thanks, Jeff. I'm very pleased to be here with all of you today, and thanks for joining on a Friday morning or afternoon. So today, my hope is the title of my talk is using epigenetics as a tool to understand firefighter health risks. And my hope today is that you will come away with an understanding of what epigenetics is, and especially how it can be related to cancer development. And then I'd also like to tell you about just a few of the analyses we've done so far with epigenetics in firefighters that are exciting. And I'd like to leave time for questions at the end too. So please feel free to ask me about any of our projects, even that I don't get to today. And everyone has probably seen this before, but I just want to frame where epigenetics falls into this paradigm. So IARC declared several years ago that firefighters are at increased risk for multiple cancers. But how does this happen? Of course, you have to think about exposures, which are kind of on the bottom right part of this graph. You know, there wouldn't be cancer if there wasn't exposures getting into the body that could lead to that. And you also need to think about mechanisms. How do those exposures impact the body in such a way that healthy cells end up, turning into cancer cells, tumors, and even eventually malignant cancers. And this is the part that my research hopes to understand. And when we think about mechanisms, you don't have to understand what all these things mean, but basically there are these 10 mechanisms called the key characteristics of human carcinogens, which are 10 very common ways that things that cause cancer work in the body to lead to that. And the one that I focus on is epigenetic alterations, which there's strong evidence that this occurs in firefighters. And there's also strong evidence for many types of carcinogenic things that they lead to these epigenetic alterations that eventually lead to the disease. But what does that really mean? And many of you here have already seen me present before, so you've probably seen this before. But when I think about the epigenome and how to describe it, I like to use a music metaphor. Most people know what the genome is. It's that code ACGTs that's in each of your cells. The genome is like a piano. It has certain keys, they play certain notes, and that doesn't really change. But the epigenome is how that's expressed. So we have, the same copy of this code in all of our cells, but our different cells use it differently. They don't express it the same way all the time. So much like you can use a piano to play thousands, millions of different songs, that genome in each of our cells can be expressed in different ways at different ages and different tissues, and also in a healthy or diseased tissue. And this is a very scientific definition in case anyone wants to know that. Epigenetics is technically what we call heritable, but yet reversible, changes in gene function that occur without changing that actual sequence. And the picture here shows a cartoon zoomed in on what a bunch of these different things are, and I'll describe some of them soon. But these are basically things on top of your genome that control what genes are being turned on and off. And it can be passed on across cell divisions and sometimes even across generations, but it can also be modified. So there's some hope when we talk about the epigenome. If you have some kind of bad coding in your epigenome, it's something that we could potentially change and make healthy again. And there are different types of these marks. There's a whole bunch of them. Today, I'm really only going to talk about DNA methylation and microRNA. But what you need to know are these are basically different mechanisms that are in the cell, making this epigenetic code that determines what genes are turned on and off. And we measure DNA methylation because it's fairly stable and it often represents the impact of a lot of cumulative exposures. We also measure microRNA because they're important for gene regulation too, but they can be altered maybe a little more rapidly after exposures. And just so that you don't come away with the idea that anything in the epigenome is bad, this is, a very important thing. We need the epigenome. We need it to function. It allows our cells to do their jobs. And, everyone here started out as an embryo, a single cell that turned into all the different cells you have in your body now. And all of these cells have the same genome, but different epigenomes. So the epigenome in a liver cell is very different than a neuron, and in a healthy cell than in a cancer cell, it's also very different. And so in healthy cells, though, we need them. We need this code, and we need it to do its job in each of our tissues. But we're talking about it today because we know that when this code isn't quite right, it can contribute to disease. And another example, a kind of visual I like to use to demonstrate this is, if you know any identical twins, twins have the same genome, but they don't always develop the same diseases. And when we zoom in and look at their epigenome it's more similar than it would be between two unrelated people, but it's not identical. And as they get older, accumulate different kinds of exposures and experiences that impacts their epigenomes, and they become more and more different as they age. And their risks for diseases are also different. One twin might develop a cancer and another one might not. And when there's ways we can look at this in the lab too. So these two mice are also identical twins. Their genome is exactly the same, but they obviously look super different, and they have different risks for health conditions too. The yellow one is not only a different color, it's also obese and it develops tumors more easily. And we know, because we use these mice in the lab, there's just one gene that's turned off in the brown mouse and turned on in the yellow mouse by some epigenetic regulation that makes all the difference here. So epigenetic differences can be very powerful. They can have a lot of impact on health and disease and any type of phenotype, but they can be impacted by exposures. And we have to look out for this. And this is why we study this in firefighters. So we know from a bunch of different studies that all different kinds of exposures, different chemicals, even psychosocial stressors can alter the epigenome in young people and also in adults. And this can be a problem because even if you have an exposure one time and say it removes some methyl groups from your DNA, then when the cells that happened to later on divide, they pass on that new pattern to the next set of cells. And then they keep passing it on and on and on. And if this has changed the regulation of a gene that you need, this could contribute to disease developing later on. And kind of bringing this back to cancer again, just to showcase how that all works. cancer is sometimes thought of as an epigenetic disease. If you compare, say, um, you have samples from somebody who had a liver cancer and you take a portion of the healthy liver and you take a portion of the tumor and you profile the entire epigenome, it's very different in the healthy cell and the cancer cells. So the cancer cells, their epigenome is now turning on a lot of genes that promote cell growth and all these other properties of cancers and turning off genes that would protect against that. And this is just another way of looking at it. Normal healthy cells can accumulate not only mutations, but also these epigenetic alterations that really impact how that cell functions, eventually leading them to become malignant cells. And this is another way that I've been thinking about it lately, and this is important for um, cumulative exposures over time when we think about how they contribute to health. You can think about cancer risk accumulating over time. In this bucket, purple represents genetics, what a person is born with. So maybe someone is born with genetic variations that make them a little more prone to certain kinds of cancers. But they're not just going to develop them just because they have that underlying genetic risk. But say over a lifetime, accumulate certain hazardous exposures and also epigenetic changes from those exposures accumulating over time. And eventually, if this bucket is full, you could think of having enough damage done to your cells that perhaps a person will develop a cancer. But, before you get to that tipping point, you could think about ways to reduce some of these hazardous exposures. And while you can't fix the genetic part, you could possibly fix the epigenetic part by having good exposures that counterbalance some of those effects that have happened. So summary so far, before I get into how we use this in firefighter research, we need the epigenome. It's important in the body. It controls which genes are turned on and off in what tissues and when. It's very important. But it can be altered by hazardous environmental exposures, and too many changes to it could lead to diseases such as cancers. But there's the potential that we can find ways to stop or reverse this process and make tissues healthy again, which is a hopeful part of this field. And when we think about where it fits into the whole disease paradigm, I put cancers here, but you could actually put cardiovascular disease and some other diseases in there as well. Really, diseases are contribution of genetics and a lot of exposures from both occupation and, your whole life and other parts of the environment around you. And some of these exposures can modify the epigenome and that can contribute to disease later on. But, there could be ways we could protect health by using the epigenome and by preventing hazardous exposures as well. And with all of that in mind, I've been working with Jeff and the University of Arizona team and the FFCCS team on a lot of projects where we ask many different questions, but they generally fall into these buckets. We ask what exposures modify the epigenome in firefighters. Do these epigenetic changes contribute to higher risk for cancer among firefighters? And that's something we're going to have to study over years of time. And are there different interventions that we can use that could reverse some of these changes? And all of this work is being done in the firefighter cancer cohort study, who I think all of you are probably familiar with, but just in case you're not, this study has the mission of conducting community engaged research with the fire service with the goal of preventing health conditions such as cancers. And we want to enroll 10,000 firefighters and follow them for up to 30 years or the duration of their careers. And the study design, after firefighters are enrolled, we collect blood and urine from them that we can use to measure lots of different things. And ideally, we follow up firefighters about every 2 years to get updated health information via survey and also new samples. And this is a snapshot of what the FFCCS looks like right now. We have almost 9,000 firefighters, getting close to that. So we're very close to our goal from 33 states and 400 departments all throughout the US. All the states in red are states that we have firefighters enrolled in. And we've done enriched recruitment for certain groups. We have a special wildland study that focuses on just recruiting wildland firefighters, a women's study that's enabled us to recruit somewhere around 1,500 female firefighters. And then I'm also leading a study where we look at firefighters who have responded to high risk incidents, which I'll talk about in a little bit. And again, these are in general how the study works. We enroll firefighters usually at their stations or some kind of central location, collect blood and urine, collect a lot of survey information on health history, occupational practices, things like that. And then we collect samples that can be used to measure lots of things immediately sometimes, and then we store them so that when we have funding or when a new question comes up, we can go back to these samples and measure things in them later on. And in this today, I'll be talking about some epigenetic marks, microRNA, and DNA methylation that we're able to measure using the blood samples. And this is another picture of what some of our questions fall into. So ultimately, our goal is prevention and protection of health. And we get at this to understand this. We want to know what exposures are impacting health, how can we reduce them? We want to know what cellular changes are happening and how can we treat that and possibly reduce that, or how could we even use that information for early detection of disease and other types of prevention or intervention efforts. And so today I'll mostly be talking about what we do with the cellular changes, but I think there's been some talks in the series so far on some of the exposures in the past. And if you missed those, I think the videos are available on the website. So this is one design that we've used to understand how epigenetic biomarkers change over time in firefighters. We first wanted to ask, does the epigenome change at all in firefighters? And we used a design that collected samples from new recruits before they'd really started their live fire training, and then samples from the same people again 2 to 3 years later. So this is a great design that allows us to see when we compare these epigenetic marks before and then after about 2 years of work, what's happened. And we can assume that if there is a lot of change, that could be from the cumulative 2 years of exposure that those individuals have just had. And in case anyone's interested, we measure something called microRNA that are these really tiny, 22 nucleotide long RNA that actually target and bind to other genes. So they turn off other genes. And we have a technology that allows us to measure 800 of them. There's 3,000 of them in most people. So we're measuring about a quarter of the ones that really exist, which is a pretty good number. It gives us a good snapshot of a lot of these important microRNA and how they're being expressed in the body, in the blood. And this was an analysis conducted some years ago by Alicia Jung, and she found that when we looked at all those 800 microRNA, there was 9 of them that had, strong changes that we were confident in with our statistical tests that changed over time in these new recruits. So from when they weren't really working to after they've been working for a while. And this included some oncogenes or cancer promoting genes that were turned up and some tumor suppressor genes that were turned down. We did a similar analysis where we isolate DNA from blood and we're able to measure these methylation marks at almost 1 million sites throughout the whole genome. And these sites, we get multiple ones per gene. So they basically cover every known human gene. And we use all of that data and we try to find genes where these methylation levels have been changed. And again, when we did really strict statistical tests, we found 680 of these different sites, some of them that went up, some of them that went down, that were different between baseline and follow-up in these new firefighters. And these sites were in all kinds of different genes, but some of the genes were linked to immune function, cancers, and also neurological function. And this was one of our first studies that really showed us these epigenetic marks, microRNA and DNA methylation, do change from cumulative exposures on the job. And they're in genes that are relevant to cancers and other disease phenotypes. And these are in people that are healthy, as far as we know, people that didn't have any cancers or anything like that at the moment. So this means the body is adapting to these exposures, and it could be in ways that could lead to disease potentially later on. And in this particular study, almost all of the firefighters were men. And we've been wondering, do we see the same effects in female firefighters, or does their epigenome respond differently to exposures and in what genes? And with that in mind, we've done a lot of epigenetics in the women's study as well. In the women's study, we first did a cross-sectional analysis comparing incumbents and recruits, these same markers, measured in the same way, this microRNA expression and DNA methylation, and just compared groups of a little over 100 incumbents to recruits at one point in time. And we found in the cross-sectional analysis for microRNA that there was 50 of these different microRNA that it says change over time, but it should be were different between the women in the two groups. And some of them were enriched in pathways that are important for autoimmune diseases and also some female-specific cancers, as well as general cancer signaling pathways. And we were able to compare this with, on the right column, the results of what we looked at in the male firefighters, and two of the 50 were also exposure associated in the men's study. So these might be just really strongly exposure associated microRNA that I want to look into further. And then there are a couple other microRNA that we found in the women that we previously saw associated with responding to a WUI fire in another group of firefighters. And from there, we took the longitudinal approach, again, with just women, looking at recruits followed over time for about 2 years, measuring the same analytes again. And we found 105 microRNA that changed over time. This included 24 that we saw in the cross-sectional analysis. And so these ones we consider to have now strong evidence that they're exposure associated, since we saw them in these two different types of analyses. And some of them were also associated with WUI fire response in the past. Some of them were ones we had identified in our male study. And these are microRNA that, we want to look into further, like what are their functions? What are they doing? Is there a way we can target them and get them expressed at their normal level again? So that's possibly work we could do further down the line. And when we look at what are all these microRNA related to, we can kind of look at what the functions are of the genes that they're targeting. And when we did some what we call pathway analyses to look at that, we found that a lot of these are involved in pathways that are linked to autoimmune diseases, female cancers, and also general immune function and signaling. We did something similar with the DNA methylation analysis, and Cynthia Porter conducted this analysis, and she found there were 17 genes that had differences between incumbents and recruits in their DNA methylation patterns. And again, some of these are genes that have been linked to female cancers before and other functions. And then when we did a longitudinal study, we found 13 of these also changed in new recruits over time. So again, we think that these ones have fairly strong evidence for being associated with just cumulative exposures on the job over a couple of years. So in kind of the big overview of the women's study is that we did see microRNA and DNA methylation that changed in women too, between incumbents and recruits, and recruits followed over time. And they were enriched in disease pathways that, we didn't pick up when we did the study in mostly men. So we see some breast cancer, endometrial cancers, ovarian cancers that might be things we want to, watch out for in future studies of women firefighters, as well as autoimmune diseases, which isn't something that's gotten a lot of study in firefighters yet either. Now, bringing it back to, what else are we doing? So in those studies, the way that exposures were measured was pretty crude. Really, it was just time, saying you've been working for 2 years. Is that associated with any of these cellular changes? But other things can happen in those 2 years too. Other things happen, aging, other environmental changes that could have contributed to that. And also, we don't know what in the cumulative exposures was important. Is it stress? Is it sleep disruption? Is it PAHs, smoke exposures, PFAS exposures? It could be all of the above. And with this kind of design, we're not really able to get at what exactly exposure was important there. But we have some other studies that I won't talk about today where we look at PFAS levels specifically and see how those correlate with some of these changes. But we also have the opportunity down the line to do that with other specific exposures as well. But one thing that I've been working on lately is, instead of thinking about cumulative exposures, thinking about big one-time exposures. high risk, low frequency incidents that are a huge exposure burden at the time when a first responder is there on the incident. And we don't really know, what is the contribution to health down the line for one big incident versus just, years and years of cumulative exposures on and off. And so for today, I'm going to define high risk incidents as things like large chemical fires, multi-day WUI fires, natural disasters, responding to building collapses, incidents that firefighters have to respond to, where they can have high dose, sustained exposures for hours to days or even to weeks. And they, might be wearing full gear like the firefighter in this picture here, or they might not. So, how they're exposed may differ by the incident as well. And we don't know a lot about how these incidents contribute to firefighters' health risks. We know a lot from the World Trade Center because there was very long follow-up studies for first responders after that helped us learn a lot about health risks from responding to that. But apart from that, there's not a lot of other incidents that have, gotten the same kind of research done. And there's a lot of things that are really important to understand about these incidents. And I haven't been able to research all of this yet, but it's really important to know on the exposure side, what are firefighters exposed to during these incidents? What actually got into their bodies versus what did the gear, protect against? Are there short-term toxicity, things that happen right after, right after or during the incident that go away and then everything's fine? And also, kind of importantly, is there any long-term toxicity or disease risk from responding to these incidents? And we worked on a couple of these now. I'll give you an example of one, but we can talk about the others in the Q&A if you'd like. And so, of course, I'm the epigenetics person. So there's a lot of ways to evaluate both short-term and long-term toxicity, but I'm using these epigenetic markers as one way to look at toxicity, both right after an incident and also a couple of years down the line. And the first incident like this we did research on was the East Palestine train derailment. And we wanted to know, did responders to the train derailment have altered epigenetic markers shortly after the exposure? And do these persist longer term? And I think people have probably heard about this, but this was now a couple of years ago. A train was transporting very hazardous chemicals. Vinyl chloride is known to be carcinogenic, as well as benzene. So this train derailed and caught fire in a rural area of Ohio. And it took a couple of days basically to put this out. And during that response, there was 350, maybe up to 400 firefighters, mostly from volunteer departments, from an area, roughly approximated by this circle on this map. They came from 3 different states to respond to this incident, not knowing, really, especially for the first set of responders that came, not really knowing what the chemical hazards were that were there at that fire. And we weren't able to get over there right in February, but we had enrollment events 2 and 4 months later in April and June, where we enrolled 62 firefighters that had responded to this incident and 26 that did not, that were from their same departments and can serve as kind of a control. 75% of our participants were volunteer firefighters. And then we also were able to go back and follow up a good portion of them, about 50 of them, 2 years later. And when we looked at the initial data, in this case, we couldn't actually measure exposures because vinyl chloride, if it had gotten into their bodies, it actually leaves the body pretty quickly. In us collecting samples 2 to 4 months later, it was too late to actually measure that. So instead, we measured microRNA to see if the body had a signature of toxicity from responding to those exposures. And we found that in the responder group, they had 17 microRNA that were different than the reference group. And this included a tumor suppressor that went down, and then also some tumor suppressors and other genes that went up. And so we don't know exactly what this means for health. Some of this could have been adaptive responses to the exposures. Some of them, might not be permanent. Some of these things might go away. So with that in mind, we collected samples again 2 years later and have those samples at the lab now to measure these same microRNA again and see after 2 years, did things just go back to normal the way they were before with these microRNA in their body? Or do they still have this elevated signature of having this response to those exposures even 2 years later. And we think that that will be important to kind of understanding if this incident has, just short or short and long-term effects for health. We're doing something similar now with several other big incidents that have happened in the last couple of years. We collected samples from about over 100 firefighters that responded to the Maui wildfires in 2023, both a couple months after the fires and again 2 years later. And we're measuring these microRNA to see if we can see those signatures of short and long-term effect from those exposures. We're also doing a lot with firefighters that have responded to the LA area urban conflagrations. And in that case, we have some firefighters who we had samples collected before because they were already in the FFCCS. So we're able to really look before and after the fires. What are some of the effects on the proteins in their body, some specific exposures we've been measuring, and a lot of different things that we're working on with that group. And most recently, we also worked with a group in Fairfax County in Virginia on a metal recycling facility fire that their department responded to. And there again, we had samples from firefighters before and after this, in this case, day-long fire. And we're looking at, what exposures went up right after the fire and also what effects did it have on some of these molecular markers in the body. So that's kind of profiling, figuring out what effects are happening. Long term, we really want to know what can we do about it. Of course, the very best thing would be prevent exposures from getting into the body. Having all of the best gear in the world that would prevent all exposures from getting into the body in the first place, decontamination procedures that can help with that and reduce what gets into the body at the end of the day. That would be the best thing in the world. But until we get to that stage, we also want to understand, are there other types of interventions other than reducing exposures that can help when the body's maybe already been damaged and help counteract some of those effects? So with that, Jeff Burgess's group is doing a lot looking at, in Arizona, looking at in some ways, general health interventions, things like exercise interventions, intermittent fasting, seeing if these interventions that have overall just beneficial effects on health can reverse some of these mechanisms, make the epigenome look healthier, for instance, in a way that will counteract what happens from exposures. Jeff's group is also looking at blood and plasma donation as a way to reduce PFAS levels in the body and trying to see if, when some of your PFAS gets out of the body, does that have beneficial impacts on the epigenome and elsewise? And then I've been working with some firefighters in San Francisco to look at whether broccoli seed supplements basically are also protective for health, reduce inflammation in the body, and possibly these epigenetic markers, again, as a way to combat, if you have one good thing and one bad thing, can they, and essentially kind of cancel each other out. So that's where a lot of this work is going, and I hope we'll be doing a lot more on this side of things farther down the line. But as a wrap up, thinking about why is this important at all? Why does this matter? We can use epigenetics in a lot of ways, on the science side, on the policy side, on the medical side. One way is the risk assessment of exposures. Sometimes there's some exposures that we can't even measure. We don't have a good way to measure in the human body. We measure things like metals and PFAS well, even PAHs. But for instance, when those firefighters in East Palestine were exposed to vinyl chloride and we didn't get the samples from them within a couple of days, we couldn't measure that. So we can use some of these epigenetic markers to look at the impact of exposures, even when we weren't able to directly measure the exposures, and to also understand the risk from those exposures, what they're doing to the body. And then we can use this information not only to help figure out what firefighters need to be protected against better, but also develop prevention or intervention strategies that may combat or reverse some of these effects before they accumulate enough to turn into a disease. And long term, again, we really want to be able to identify the most harmful exposures and target them for reduction in firefighters. We want to make biomarkers that are really, really strong that we can use, that we can measure real easily in a blood sample, for instance, that can warn us of toxicity and cancer risks in an individual person. And we also want to use this to inform intervention strategies that could protect health long term. So the take homes for today, hopefully after today, you've learned that epigenetics is an important mechanism in our cells that is essential to all biological function. But the epigenetic can be majorly damaged in disease states like cancer, and exposures over time can contribute to that damage. And we want to use this knowledge to understand health risks and eventually protect against them. And I want to acknowledge the funding that we have from these projects. I only talked about, these are all basically different sub-studies of the FFCCS that have been marching along over time and kind of resulting in the 8,000 person cohort we have today. And there's, dozens, hundreds of people that are, I wish I could list all here, that have been contributed to, you know, all of this work that I talked about today. And we have so many fire service partners who we could not do any of this without, that have, volunteered their time to be in the study and also to help us recruit others into the study. And we really couldn't do this without you. So I want to acknowledge all of our scientific and fire service partners, including the ones now on this slide. And with that, yes, now we have a lot of time for questions.
Perfect. So, please either just raise your hand so Jackie can call on you, or you can put a question in the chat. But we scheduled this specifically to allow enough time to answer your questions. And Jackie presented a lot of great information. So while we're waiting for folks to chime in, Jackie, do you have any idea? So you kind of mentioned some of the interventions that are in place. Do you have any idea when we'll be able to kind of share information, for example, on the sulforaphane study?
For the sulforaphane study,
The
broccoli, yeah, that's the broccoli seed, is how I described it a minute ago. So the broccoli seed supplement study, we are preparing the report back of our results that will be shared with the participant firefighters from San Francisco, I think in the next couple of days. So yeah, after that, once they receive it, I think that's something that, we could put on our website or share with all of our fire service partners as well, the results of that. And then we'll be publishing it after that too. So yeah, those results will be coming out very soon. And that's something we might do more work on in the future as well, because that was just a small pilot study to start.
Awesome. Thank you. All right. Questions from the group? Why don't we stop sharing the slides for just a second so they can kind of see each other a little bit more there. Perfect. Looks like there's a question from Ryan, Jackie.
Oh, Ryan. Hi, Ryan.
Hey, good afternoon, everyone. Hey, I know I reached out to you personally about the sulforaphane study, but as you consider the next phase of that, are you going to be looking to expand out to other departments or just expand there in San Francisco?
That was a great question. I haven't really planned the next steps, but I think it would need to involve more people if we do more with that. So I'm sure Fairfax could be involved if you would like, but Fairfax County.
I would love that.
Yeah, because we just had a small study. It was, really about 50 people that took the supplement, 50 that didn't, and over a fairly short period of time as well, 3 months. So I think that's something that, could be expanded out over a longer period of time and with more firefighters eventually.
Perfect. Thank you.
And Ryan, I think something unique about that is that San Francisco has that union-based cancer foundation. I'm sure you know a lot about it, but just for everyone else on the group, they actually put funding in for this to be done. I think defrayed the cost somewhat, but they had a substantial investment to kind of make this happen. And I think that they also got the producers of the rock shot to provide it. And I think just one other thing is this is not a randomized control trial. So Jackie, I don't know if you want to kind of talk about the differences between an RCT and one of these observational trials, just to kind of speak about the limitations of this as compared to other study designs.
Yeah, sure. So when we're testing something like a supplement, or they do this in like the pharmaceutical industry too, the gold standard to test whether something works or whether something causes something else to happen is a randomized controlled trial for people. So in this case, that means you take 100 people, or probably really more, then you randomly assign them who's going to get the supplement, who's not, and then usually the person that's not gets some kind of placebo, another pill, so that they think they're getting it but they're actually not, kind of thing. And then you follow those groups over time, and if say there's a positive health effect in the supplement group, it's a good design to show yes, the supplement caused that change. But in what we did, this was just a small pilot, and there's a lot more kind of like oversight and special kinds of ethical approvals and other things that go into a randomized controlled trial. So we decided for this first pilot just to do an observational study. Basically, the supplement, the Brock shot, was offered to a whole group of firefighters, and then some of them decided to take it and some of them didn't. And so it wasn't random. And we just followed, okay, we just compared the group that took it, the group that didn't. But it wasn't a trial because we didn't assign who would get it. They just decided themselves who was going to get it. But oh yes, Ryan.
Sorry. Question then. Are you able to say what biomarkers you were looking at? Was it like HSCRP or IL-6? What were you, and future consideration of other biomarkers, I guess?
Yeah, and again, since it was a pilot, we just measured a couple of things. We measured CRP, so can be a marker of inflammation essentially in the body, but that was the only inflammatory marker we measured. Recently, we also measured glutathione and another marker of oxidative stress in the body. And then we just got those data back though. And then I also measured epigenetics, some like the DNA methylation that I was talking about today. So with that, I can kind of look at how a whole bunch of genes were impacted by this. But yeah, that's all we measured for now. But I think other inflammatory markers would be neat to measure as well.
Thank you. That's really helpful.
Mm-hmm.
And then there's some comments here in the chat, Jackie.
Yeah, I see those. So Heather has a question. Let's see. Yeah, so she's asking, when we compare recruits and incumbents, could some of the differences we see just happen with age? Yes, that is always a big study design problem when we're comparing recruits and incumbents. Usually the incumbents group is on average 10 years older or something like that. And there are epigenetic changes that happen naturally with age. And we kind of know what they are too from other studies. So that's why I like to pair this with the recruit designs too, because when we follow the recruits, it's only over 2 years. So there's a little bit of age-related change that can happen in 2 years, but not much. So if I see it in the incumbents and recruits and also in the longitudinal design, then it's more likely to be exposure related than just age related. And then we have another question about blood and plasma donation. This is a great question. This question always comes up because it's really important. So if one day we get to the point where we say we're recommending blood and plasma donation for people with PFAS in their blood, which in some ways that's already being recommended now, is this a problem for the people that get your blood later, is that a threat to the public? And so I know there's been discussions with the Red Cross about this and other organizations, and right now the what people are thinking now is that the individuals that need to receive that blood are in such dire need that getting the blood is still better than some level of PFAS contamination that they may receive from that blood. So right now there's no screening at normal blood donation places and things like that for PFAS levels. You know, if you're PFAS exposed, your blood could still go to somebody else. So, you know, that guidance could change possibly in the future. I know there's also a group working on developing some kind of technology that could just destroy PFAS, like in the bag of blood somehow. But I think that would be some years before that would work. But yes, that's a really important consideration. Okay, and we have here, I'll take Derek's question and go back to the chat.
That was just a comment off of that. So we just had about 1,085 of our members get their PFAS results. And the people, sorry, I'm in a restaurant right now at work. The people that were really elevated, they already reached out to Red Cross to do blood or plasma. And Red Cross made a change last month that you can only donate plasma if you're AB blood type. Nobody else is able to do it as effective last month, at least for Red Cross. So I'm not sure if there's other places that you can donate plasma that are Red Cross affiliated and they're okay with it, but this is a change that just happened to Red Cross.
Jackie, can I try that one? So I think Derek, thank you for sharing that. And that's important for us to know. Plasma donation looks like it's going to be, you know, as the Australians reported, be more effective than blood donation. So as far as I know, and this guy Sean's got his hand up too, the for-profit plasma locations will still be taking, you know, any type. But Sean, you had something to say on that?
Yeah, basically that is correct. So we initially Red Cross, they, you know, it's interesting that they fully went that way. When we were initially talking to them, they do have preferences when it comes to plasma donation. But again, we were able to kind of bypass that for at least our group that we were kind of pushing through. So that's interesting across where you guys are seeing that that's now fully in effect. But the for-profit, you know, your CSL, your BioLifes, Vitalent, you know, other for-profit locations don't have a restriction on blood type for the plasma donation. So there are other options that they can do that are not Red Cross.
Sean, is there any way you could send me the list of all those ones? So I need to send something out to all these people that are thinking about it just so they don't get frustrated or waste
Yeah, I can get you the ones that we know of. Okay. Every state is a little bit different on which what their plasma facilities are called, and I don't want to say just the state. There's also regions, but we'll get you the ones that we knew were in your guys' region for Southern California.
And yeah, thanks for bringing that up, Derek. That's a very recent change. It's really important for us to know. So Tim has a talk about, oh, this is a good question too. So in our recruits, when we see changes 2 years after, you know, working for about 2 years, when we look for a longer amount of time, do we see basically even more changes or not? And that's a question we're still exploring because now we're finally getting to the point where now we're able to follow up some firefighters, you know, 4 years out, 6 years out. We're really just starting to do that now, and we can look in the same people, hopefully, you know, longer over time. But just by kind of estimating this using firefighters' reported years of service, it usually the impact seems to level off in a way. I do tend to see more changes in a new recruit when they went from not working to working for a couple of years than in, you know, a firefighter that's worked for like 1 year or like 10 years versus 20 years or something like that. So it does seem that maybe like being new to the exposure, you know, might bring on like a certain set of changes, and then maybe a smaller amount of them can kind of continue to accumulate after that. at least in what we're measuring. We can only look at blood, so we have no idea what's happening in other tissues. We can only use blood as kind of a surrogate of everything else. And then Neil has a question in here too. So some compensation systems require demonstrating that you were exposed to chemicals. And yes, it's very hard when the exposures were sometimes even days ago to be able to quantify and show that when you need to. So yeah, I do have a hope. We're not quite there yet, but I do have a hope that we could have these epigenetic signatures that we could say this person, like we take your blood and we measure this and be like, okay, because this set of genes looks like this means this person was exposed to, a lot of PAHs in the past or a lot of PFAS in the past or something like that. So that there's outside of firefighter research, there's a strong signature like that exists for smoking. You can still see, even when people have quit cigarette smoking, you can see this signature that they were exposed to it in the past. So I am really hoping that we can develop really repeatable signatures like that for some of these other exposures in the future too, that could be used kind of like retroactively.
Thank you.
All right, are there any additional questions for the group? All right, well, while we're waiting to see if there's any final questions, I'd just like to make the observation about how far we've come in the last 5 years. IARC used this information to help support the determination that occupational exposure as a firefighter causes cancer. So Jackie was there presenting her research as part of this group, along with with others, and so that's it's already been really influential. But back then, it was really determining that firefighting exposures cause cancer, and now we're I think progressed to the point where we're able to look at individual types of exposures, individual firefighter groups, and really starting to look at interventions. So I mean, this has come so far in the last 5 years. It's rather remarkable, and I just foresee continued use of these markers to help us provide information to firefighters about ways they can reduce their risk, not just of cancer, but other things like reproductive and neurological as well too. More and more information on that. Chinese is a really important area. So I just want to acknowledge Jackie's contribution to moving us all forward collectively.
Yes, thanks, Jeff. It's been a great team effort. Yeah, I'm really excited about what else we'll know 5 years from now. I think it'll be even exponentially more.
All right. So any final questions from the group?
Dr. Burgess.
Oh, please go ahead.
This is Scott Figgins. Hey, I just want to reiterate what you just said. Thank you to Jackie, yourself, everybody. I know you say it's group effort, but there are key people and you guys are them. You have truly saved lives and helped individuals across the country. I certainly know in the state of Arizona, several individuals would not have been approved for occupational cancer had it not been for your work. So just want to reiterate that point and say thank you.
Oh, thank you, Scott. And I would just say that, again, Jack has been, leading us on this effort. But as you said previously, it's a community engaged effort, and we would not be able to do this without the incredible support of the fire service and the liaisons with each of the departments we're working with. Because again, as Jackie said, this is a team effort, and the fire service is a absolutely critical part of that. Sorry, Jackie, I should have let you say that.
No, that's exactly what I would say. But yeah, thanks for sharing that, Scott.
All right. Well, thanks everyone. Appreciate it. And again, this will be recorded if on the website. If anyone would like to share it with others. Again, looking forward to the next session in about 4 months from now.
All right. Have a great day.